Hello there. My name's Dr. Frank Abernathy. I'm trying to wake up here. I need another cup of coffee, but I'd like to talk about something that's not supposed to exist. An inconvenient truth, you might say. Uh, I'm a I'm a chromosome person. I like to study chromosomes. I haven't studied them, been in the laboratory studying chromosomes for about 30 years now. But I still like them. I've always wondered, that they're kind of magical to me, and I wonder how in the world they operate and how they're put together. I've always wondered that for many, many years. And so I worked on that at Ohio State for, I got my PhD there. And I am still fascinated with them. They're very fascinating structures. And I would like to share some of my fascination with you if you would allow it. So what I'm going to show in this particular video is some rubber band models which I've used to try to explain how I think these things are actually put together. And here's one of these rubber band models. And if you'll notice, it looks it looks like a funny looking rubber band because the reason it looks this way, I cut it. It used to look like this. It was two rubber bands, and I cut them in the middle, and then they formed this thing, which has these, I call these dimples. And I'm going to talk about this a lot more in the video. And if you just take a little time, it's only I'm going to try to keep it down around 10 minutes. But I think you might enjoy it if you have some time to watch it. So here we go. How are our chromosomes really put together? Why does it matter? Well, chromosomes contain DNA, the blueprint for our bodies. You may have heard the old phrase, or you may not have heard the old phrase, ontogeny recapitulates phylogeny. And what that means is that if you look at embryonic development, from fish all the way up to humans, if you look at it very carefully like is shown in this chart, you will see that in the early stages, at the top of this chart, you can't really tell a fish from a human, or a salamander, or a tortoise, or a chick, or a pig, or a cell, or a rabbit. All that is determined by their DNA. So it's very important to understand how the DNA operates. You know, if a mechanic wants to fix a car, he needs to know as much as possible about what makes it tick and what can go wrong. That way he knows what tools to use and what tools not to use. The same thing applies to the treatment of genetic diseases like cancer. Understanding basic DNA structure isn't enough. We also need to understand how DNA is put together and functions within the confines of a chromosome. This is a typical textbook chromosome model. If you'll notice, this whole thing is a single strand of DNA. If you unravel the entire chromosome, it's supposed to be one single strand of DNA. Well, actually, that chromosome has been duplicated, so it would be two strands. So how does that chromosome model explain what you're looking at right now? How in the world can it explain that with a single linear strand of DNA? I'm going to show you a hypothetical rubber band model of a small section of coil chromosome. Now I know that looks kind of odd and funny, so you have to really use your imagination here and think of that like you took a hot dog, the hot dog part of the chromosome, and cut a chunk out of it. And so that is a chunk of it right in the middle. You'll notice that the rubber band itself is considered in this model coil chromatin fiber. It's a fiber, which is mostly made up of protein and DNA. And the paper clips represent chromatin, which has been further compacted with more protein. And we'll show you what I mean by that in just a second. Okay, the chromatin relaxes, revealing 4 beads. So you see 4 beads there, each one of them compressed by the paperclip protein. And there's a large circle there with 4 beads attached to it. those beads light up under the light microscope under UV light with a fluorescent dye called acridine orange. And there's an example of this on the right hand side. You see an actual circle, an actual photomicrograph, which has about 50 of these beads around the circumference of it. It looks like a perfect circle because these things are small enough. They have similar charges. They will repel each other and move away in an aqueous environment to create a circle. That one, the original image, was at 400x magnification. Now let's do a bead protein removal, and let the bead proteins, once they're removed, allow the beads to relax. Here's a fully relaxed, non-beaded circles. You see a big circle with 4 little circles surrounding it. They're glued together with super glue. Fully relaxed chromatin reveals that large circle. Now, in actual photomicrographs at 400x with acridine orange and fluorescent dye, you would never see those little circles when they're fully relaxed. DNA is just too small to be seen at that magnification. If you want to see that, you're going to have to isolate the DNA and look at it directly under an electron microscope, which we have a sample of that on the right, of what looks like, for all manners and purposes, a DNA flower. But that magnification is at 125,000x. Alright, we're going to, for the sake of argument, cut one of these beads where it connects, or where the bridge connects to the large circle. You can see where that's been done by the yellow arrow. It generates 2 dimples, as it shows in the second diagram. And I'm going to show you examples of these dimples and bridges in the photomicrographs below these. You'll see a dimpled chromatin at 400x magnification on the left, and on the right you'll see another dimple, but that dimple is in DNA circles. And again, that magnification is 62,500x. So let's go on and cut another circle, and we create 4 dimples. And then we'll just go ahead and cut all 4 of them. You can see where they've all been cut, and it creates that giant dimple circle that you see on the right. If those circles were cut more than once, you would cause a piece of that dimple to come out, and DNA information would be lost, and then that would cause a permanent alteration in the DNA structure. Now you can see photomicrographs of giant chromatin circles at 400x again at the bottom of this diagram. Those 2 circles are irregular shaped because they are so large now that the electrostatic repulsion no longer applies. Now this is the 2-cut model I was just talking about. If you look up at the model in the upper left-hand corner of this diagram, that where it says tetrameric base pairing, that's supposed to represent 2 DNA circles, or replicons they're called, and they are all 4 of those strands. There's 2 strands at the top and 2 at the bottom, and they're all pairing up. Some of them pair with the original DNA strand. Some of them reach down and pair with the strand below it. And that's the kind of bonding I'm showing with that rubber band on the left. Now the 2-cut model, you'll notice those 2 arrows, it's cut on 2 separate spots, and that reveals a fragment that comes loose that you see on the 3rd diagram, and it shows it at the top as well in that diagram. Examples of these kinds of models, if you look down, you see a DNA circle with bridges. There are a number of these that can be found on my blog, but I just put one on there, and you'll see that there's bridges between those circles, and that is from purified DNA, and that is magnified 175,000 times. You wouldn't be able to see anything like that under the light microscope, only the electron microscope. And on the right you will see a dimpled chromatin. Again, this is at 400x. And you will actually see a bridge that is beginning to separate as the dimples are forming. Okay, in this next slide, we're going to show that the chromatin and DNA appear to have a hierarchical size range. A size range goes from giant chromatin circles, as you see in the third slide, all the way down to almost non-visible or below the level of the 400x magnification. In the other slides as well, you'll see all sizes of these kinds of beaded circles. And these circles, you can even find them at the DNA level when you look at the electron micrograph below the center photomicrograph. It says purified DNA. That image is at 51,500x. It shows a number of circles. Some of them are also bridged. So these data strongly indicate a hierarchical nature to the structure of chromosomes. Also, you will find that chromatin circles also appear capable of changing shape and releasing substructures. If you look at this fairly complex photomicrograph at 400x again, but not under UV light, phase contrast, you will see a variety of these different circles, and they all have different shapes, and some of them seem to be bleached out, which means they have lost some kind of substructures. You can see the substructures trying to come loose in figure 39, and as these things, as you look at the ones on the bottom, they actually seem to be turning into chromosomes. So this is something that probably is cell cycle dependent. What happens next? To the best of my knowledge, there has been no published follow-up to the work presented here. Much of these data has been published in my PhD dissertation at Ohio State University 30 years ago. I would like to see this work continued either by myself at a local university or as a non-paid mentor for a graduate student anywhere in the world. I believe that if adequate funds were made available, it might be possible to work out a deal with a university or research facility to continue with this work. This would require access to pre-existing laboratory facilities and equipment which may or may not require funding. It would require funds for disposable items like tissue culture flasks, pipettes, and chemicals. It may require funds for hiring a graduate student or postdoc unless one is already available and requires a project. You may contact me directly via email at fabernathy@sbcglobal.net. You can also check out my blog, which includes access to my PhD dissertation, manuscripts, microphotographs, models, and references. And there are the links. I also have a number of other videos up on YouTube as well. You can look for cryptic chromosome structure. Thank you for watching this video. This channel can be found by typing cryptic chromosome structure into the YouTube search panel, not Google. The companion video to this one is entitled Chromosome Video 2018. It contains detailed models of how chromosomes may have been put together using circular DNAs.